you everyone for being here this morning. I'm very excited to introduce Peter Crockford, our seminar speaker for this week. And Peter and I met initially as graduate students. He was doing his PhD at McGill University with Boz Wing and Galen Halverson. If I mess anything up, you can interrupt me. Then he moved on to do the Agron Geobiology postdoc at the Weizmann Institute with Itai Halevi, and also working in part at Princeton with John Higgins, and that was on calcium magnesium isotopes. And then he did another postdoc, partially at the Weizmann and partially at Harvard with Dave Johnson. And finally, now he is at Hui doing the Hui postdoc fellowship, which he will complete this year, and then move on to a faculty position at Carleton University in Ottawa. Peter is primarily an isotope geochemist studying neoproterozoic and very ancient environments, but it's kind of hard to put him in a box. I was looking at his CV and realized I'd say you definitely worked on more isotope systems than any of us here. So a lot of different experiences. He's also just a really great person and mentor. So I would definitely recommend to any students thinking about next steps, definitely reach out to Peter. He's amazing. And with that, I guess I will let Peter take it away.
Right. Thank you. Yeah, thanks for the introduction. And I'm just gonna move this. You can hide all this stuff. Okay, there we go. Cool. I was just talking to Matt in the hall, and he actually reviewed my PhD thesis, so I'm a pretty well-vetted speaker. So today I'm going to talk about sort of the main theme that's run through my research over the past almost 10 years now since I started my PhD, which is looking at primary productivity through time. And although a lot of my work's kind of grown into things about like carbon burial and export and things like that, this has sort of maintained my interest, and it sort of gives me the best type 45. So that's what you're getting to do. Okay, so there we go. So here's a cartoon version of Earth history, which has been sort of the achievement, I would say, of like hundreds and hundreds of studies, years and years of work for this sort of broad picture of the last 4.6 billion years of the planet. So we know some things, for example, like CO2 has dramatically changed in the atmosphere as the sun's gotten slowly brighter over time as hydrogen's converted to helium, and the density slightly increases, the pace of nuclear reactions increases, and we experience that as an increase in brightness. And it seems that the atmosphere is compensated by a steady decrease in CO2 over time through silicate weathering. Another major achievement has been looking at the history of O2, going from nearly none, or maybe none, completely in the Archean, a large jump around 2.5 billion years ago, maybe lower oxygen levels in the mid-Proterozoic, and then another jump as we move into the Phanerozoic. And ultimately, this atmospheric record is projected onto the biosphere, or maybe influenced by the biosphere by changing types of metabolisms, types of ecosystems, and all of this is projected in the ocean through changing redox environments, seawater chemistry, pH, etc. But underlying records like this is is fundamentally geochemical records, okay, which really forms the base for a lot of these pictures like this. So what we're looking at here is just a stable carbon isotope record, carbonates through time. And the main things to point out is, people have interpreted things like these positive values. Sorry, I forgot my wand. Go. These positive values here and here as maybe increases in organic carbon burial, which ultimately led to O2 rises in the atmosphere. Okay, but I think that there's still some really basic questions that we're left with when we're studying Earth's past. For how much carbon has ever been fixed? How many cells have ever existed? What has been the most abundant type of organism through time? What impact have extinctions had? And where's the majority of the uncertainty in these sorts of pictures? And so I think these are just some more fundamental things that are sort of fun to spitball about at the pub, but this is going to be largely the topic of today's talk. So just briefly, like a picture like this is probably familiar to many people. One of the major drivers of biogeochemical cycles on the planet is organic carbon fixation in surface waters of the ocean. We export a portion of that carbon to deep waters, and then a certain amount of that gets transported to the seafloor and buried for millions and millions of years of time. Okay. And the focus of a lot of my work has been trying to find tools that we can apply to ancient sediments and get some constraints on these features as we go back in Earth history. So triple oxygen isotopes for primary production. We've been doing a lot of work lately at HUI, looking at modern barium cycle to see if barium could be an interesting tool to take back into the geologic record. And then looking at calcium magnesium isotopes as a potential way to filter carbonate records to get at an estimate of organic carbon burial through time. All right. So today I'm going to focus just on primary productivity though. So again, this is the conversion of inorganic carbon to organic carbon. This sort of bridges the inorganic and organic world. And every year on Earth, about 250 billion tons of carbon is fixed through oxygenic photosynthesis, with about half of that occurring in the oceans and the other half occurring on land. Now, if we think about this back through time, I think there's some intuition maybe a lot of folks have in this room. So if we look, for example, at like a modern census of the biosphere today, and we just look at raw biomass. Okay, so if I take all life on the planet, I suck out all the water, I just put the dry carbon on a scale, where does it all sit throughout the biosphere? And this is work from colleagues of mine at Weitzman. And we can see that land plants today represent the majority of this material. Trees are heavy, but bacteria may be less intuitive, or the second biggest chunk of this diagram, because they can live so deep in the Earth's surface, right? You can tell everybody about and occupy even ourselves. We're about 1-to-1 bacterial cells external to us versus our own human cells. But ultimately, this modern census translates to some flux of CO2 in and flux of O2 out. Now, if we go back to the Proterozoic, where we didn't have some of these types of life, can we just calve off chunks of this diagram? So we get rid of land plants, we get rid of animals, we get rid of fungi, and are we fundamentally left with less life on the planet? And does that less life on the planet translate to a smaller flux of CO2 in and a smaller flux of O2 out? So that's just a prediction, that at least I had in my head when I got into this. Let's see if that might be true. So measuring productivity today is an achievement of lots of different ingenuity and a lot of hard work. So we can use satellite measurements, looking at chlorophyll content, surface oceans. We can do shipboard incubations, looking at gas fluxes and cellular growth curves and things, and project that out into a sort of puzzle piece that together as for a picture of the whole planet. But if we move back in time and we get rid of all of our research vessels, we crash all of our satellites, it becomes a really tough problem. So one place we can look, which is pointed out for the past 60 odd years by folks like James Lovelock in the 60s, which is that if you just look at the Earth from above, you can tell, or at least surmise, that there's probably a living planet. Okay? If you look at concentrations of gases like O2 and methane, these are fundamentally a disequilibrium. Or put another way, if we were to kill off all life on the planet, or maybe like accelerate our efforts a bit, but if you were to do that, methane would quickly react with O2, the methane concentration in the atmosphere would dramatically shift. O2 would react with reduced species on the surface, and its concentration would evolve to quite different levels through time. Okay, so all that's to say that we can look to the atmosphere as an imprint of life on the planet. Now, what I've been doing, as well as Anna, is looking at the isotopes of O2. Okay, so, and use this as a way to back out what this flux of O2 should be to the surface environment through time. And because oxygenic photosynthesis dominates primary productivity across Earth, this gives us an approximation for other features of the planet. How much life could the planet sustain? These sorts of questions. So I'm going to talk a little bit about isotopes, but over the years, I've sort of whittled this down further and further to keep as little of this out because it's, it's just a good way to simplify it. But anyways, oxygen has 3 isotopes, 16, 17, and 18. In order of abundance, 16, 18, 17. We present this in delta notation just so we reference all of our values to a standard. So 0 means the same thing to everyone. And we do it in parts per thousand or per mil. So we're dealing with whole numbers. That's the main takeaway. Now, because oxygen has 3 isotopes, we can calculate delta 18 values, looking at 18-16 ratios, or delta 17 values, looking at 17-16 ratios. Okay, now a cool feature was found when people started to measure this in substances. It's back as far, I guess, as the 50s. And it was cool, actually. I was at the University of Chicago yesterday, and someone gave me one of Yuri's old notebooks. So I was digging through that, and it was pretty fun to read about things like people like Emiliani being like, Dr. Yuri told me to do that. It's kind of cool. Anyways, so if we look at the oxygen 17 versus delta 18 values on a plot of virtually all substances on Earth, they plot along a line like this, okay, with a slope of about 0.5. So all this to say is that when we have some sort of fractionation with some reaction, if we know what the oxygen 18 value is, we can predict what the oxygen 17 value should be. Okay, and this led a lot of researchers in early days to suggest, like, why even measure these really, really tough, low abundance isotopes? All the information we need is actually locked into these heavier isotope ratio values like 18-60. Okay, and these are so-called mass-dependent effects, okay, where the physicochemical properties of substances ultimately dictates the isotope fractionations we observe, and that translates through all isotopic mass across an element. But an interesting set of observations was made in the 1970s by Bob Clayton, also at the University of Chicago. And what he found was he was measuring meteorites. And what he found was that there was these, all of these meteorite samples seemed to plot along the line of a slope of 1, which is very different than this mass-dependent value, which was seen in basically all terrestrial substances. So the initial explanation for this was maybe we're dealing with some other oxygen reservoir out in space, which is giving these very different values. It's subject to lots of different processes that we don't actually have to worry about on Earth, which gives this interesting slope of 1. But a former postdoc out of that lab, Mark Tiemens, when he took on a job at UC San Diego, performed a series of experiments which brought some new insight into these weird isotope effects. And so what Mark did is he took O2, sparked it in a box, and created ozone. And then he measured the triple oxygen isotope composition of the ozone and the residual O2. And what he found is that the ozone produced starts to get isotopically enriched relative to this mass-dependent line in O17. And the residual O2 becomes mass independently depleted in O17. And this seemed to bring to light a new type of isotope effect, a so-called mass independent isotope effect. Okay, and so this really excited a lot of people because it was a sort of a weird signature that maybe we could apply to other parts of your system to trace interesting processes. So sure enough, oh, sorry, that was 5 minutes. So the way that we define these triple oxygen isotope signatures or mass independent signatures or OMID is simply we measure our oxygen 17 value and we just compare that to what we would predict from measuring an oxygen 18 value alone. Okay, and if we see a deviation from this mass dependent slope of 0.5, then we have a mass independent effect of some magnitude. So with this discovery, people went out and started measuring oxygen sparing species throughout the environment. And what they found was basically every atmospheric gas, whether it's ozone, nitrate, CO2, peroxide, sulfate, CO2, all of these things plot positive, meaning they have more oxygen 17 than we would predict from pure mass dependence. And the fact that all these gases plot positive means something has to be bearing the negative brunt of these depletions for isotopic mass balance. And sure enough, the answer was O2. But even though things like ozone can have these huge enrichments, so this is in per mil, the scale here, so you can have these triple oxygen signatures up to 40 per mil in ozone, or tens of per mil in CO2. The reason O2 can have this small negative signature is just because there's so much O2 in the atmosphere relative to these other gases. All right. So thinking deeper into these observations, folks like Yakiani at Caltech started to think about what actually controls the magnitude of these signals that we see. And so he developed these simple photochemical models and developed these photochemical reaction paths to predict that the magnitude of the signals we see in ozone or CO2 and O2 is actually dependent on the concentrations of these gases in the atmosphere. And the main insight was that carbon dioxide, the amount of that in the atmosphere actually plays a large role in how negative that residual O2 value can get, because it can hold on to these heavy oxygens after they're broken up photochemically in ozone. And so all of the prediction in this paper was like, there should be a sensitivity to CO2 levels and the magnitude of the triple oxygen isotope signature that we see. And sure enough, work out of the Tiemann's lab confirmed this experimentally. So on the x-axis, we have increasing CO2 levels. And then you can think of it as the separation between the blue and red dots is just an increasing degree of mass independence. And then finally, some work out of Michael Bender's lab at Princeton and other folks applied this to natural samples. And so what we're looking at in the little picture is a picture of ice with little bubbles in it. And these are results from the Gisby ice cores from Greenland. And what they measured here was the CO2 concentrations and the coeval triple oxygen isotope composition of O2 in those same bubbles. And what they found is a similar relationship. Of course, it's messy, these are natural samples, but increasing CO2 with increasingly negative triple oxygen isotope signatures in O2. Or another way we can think of this is here we are in the glacial and here we are in the interglacial. So all of that is to say is that there's a relationship with the magnitude of the triple oxygen isotope signature we see in O2 and the ratios of gases like O2 to CO2 in the atmosphere. Okay. So putting that together, we have photochemistry in the stratosphere, which is driving this unique isotopic signature. And then this fluxes down to the troposphere and sets the tropospheric composition. That's all we had to worry about. We see these, we wouldn't really have to think about anything else in the system, and the troposphere would basically approximate the stratospheric value. But we also have this biological flux of O2. And because during photosynthesis, the O2 produced is grabbing its oxygens from water, which has a Cap17 value of 0 by definition. What ultimately sets the troposphere is you have these photochemical reactions in the stratosphere producing this unique triple oxygen isotope signature. This gets diluted by this biological flux of O2, which has a value of 0. And so it's really the push-pull between these two processes which sets our tropospheric value. So we have this interesting signal. What do we do with it? So there's some predictions that we can apply for triple oxygen isotopes and O2 back in time to see if there's any information here we can. So predictions are we shouldn't see an appreciable triple oxygen isotope signal or negative one to be specific in atmospheres with no O2. Because these signals are reliant on photochemistry involving ozone, we shouldn't see any signatures, for example, in the Archean. If O2, CO2, and productivity is changed, then this should be reflected in the triple oxygen records of atmospheric O2. And if we know 2 out of 3 of these variables, O2, CO2, and productivity, we should be able to solve for the third one. All right, so the first problem we run into is the ice core record at best goes back about 2 million years. And even though today people are working on drilling programs in Antarctica, they're uncovering maybe 3 million-year-old ice. These are unhinged ice blocks. They don't give us complete records. And we're still missing an order of magnitude here for like deep time type people. So we need another reservoir of oxygen or another archive of oxygen that we can exploit. So one place I've been looking is looking at sulfate. So here's a simple picture of, we have O2 that's coming down with these photochemical reactions, mixes into the troposphere, and then some of that O2 is going to react with sulfides on the continent to produce sulfate. And some of the oxygens in that sulfate derive from O2, the rest coming from water, which should have a triple oxygen isotope value of 0. So we can think of sulfate as a diluted version of our atmospheric signature and correct for that. Okay. So putting that all together, if we go from the stratosphere on the y-axis, we just have our triple oxygen isotope values from positive to negative. We start in the stratosphere with these really negative values in O2 from photochemical reactions. That gets diluted in the troposphere because we have this biological photosynthetic input of O2. Then that O2 can react with sulfides on the continents to produce sulfate, and that'll have a diluted version of this atmospheric signal. And if that sulfate can be protected against subsequent sulfur cycling, which can permit some oxygen exchange with water, we may be able to preserve these signatures in the geologic record. All right, so now we need a bunch of sulfates. So a lot of my work over the past number of years has been to try to find evaporite deposits, barite deposits, things from across the globe to build up this record, which has led to some really fun field work, but also a lot of great relationships with really generous collaborators. And there's still a ways to go, but we're doing pretty well in our sample suite. Some of this work's been pretty thrilling. I'm not a big climber, nor do I like heights, but here we are on the cliffs of northern Baffin Island, and I'm holding the camera, shaking. And here's just a picture of like a sort of 30 centimeter sized pod of gypsum, what's called the Angmac Formation. So this is about 1 billion years old. Okay. Now here's my colleague Marcus Kunzman, and focus on him because here he is again on the lower left here, down here. And now Marcus is sitting, I'm also holding the camera again, at the base of about a 100-meter thick sequence of evaporites in the Mackenzie Mountains, what used to be called the Gypsum Formation and is now called the Tenstone Formation. And this formation can actually blow up to about 500 meters thick in some places. So this is just to show the diversity of how these evaporites can look in the wild. So here's what the record looks like as of today for triple oxygen isotopes in sulfates. And I'll just take some time to walk you through this. So in the top dotted line here, we have the composition of modern seawater sulfate. This is work by Anna in one of her early PhD papers. On the lower dotted line is what we have for modern tropospheric O2, has a value of about -0.5. Some of your eyes might be drawn towards these really negative values that actually start to drop below. I'm not going to talk about this much today, but just briefly. So this is associated with the second snowball Earth event in the Neoproterozoic, the Marinoan. This is actually a big deal at the time because there was a lot of controversy when snowball Earth stuff was coming out. Was it a slush ball? Did we have a big water belt? A lot of the criticism leveled about the whole Earth being frozen came from biologists really struggling to understand how the biosphere would survive through these intense glaciations. And so what Hui-Ming Bao did at LSU is measure the triple oxygen isotopes of barytes deposit right after the second snowball Earth glaciation and found these massive depletions. And the cool thing was this seemed to provide some support for a hard snowball scenario. So you can imagine when we lock the Earth in ice, we're reflecting so much sunlight back to space. It's an incredibly stable climate state. And the only way to get out of it is just huge amounts of CO2 in the atmosphere. So the idea is that in a hard snowball world, you can prevent surface reactions with that CO2 and build up these crazy high concentrations. And because a prediction of these triple oxygen isotope systems is that there should be the sensitivity to CO2, these really large depletions were evidence for really, really high CO2 to deglaciate the planet. So that was cool. But I'm not going to talk about that anymore. You also notice that there's a lot of diamonds and a lot of circles, not as many squares on this. So the diamonds are barytes, the circles are evaporites, and the squares are carbonate-associated sulfate. The reason that there's not as many measurements on that is just because it's more arduous to make a cast measurement. And sometimes you dissolve rocks, you don't get enough stuff, it's frustrating. So the places to look were evaporites and barytes first. All right, so the rest of the talk, I'm just going to walk you through 4 quick vignettes of just how some highlights of this record, information I think is added to our understanding of Earth's past. All right, so the first one is looking at Earth's oxygenation. So what I have here is, again, triple oxygen isotope values on the y-axis. That's just going to continue throughout the talk. And what we're looking at is Archean sulfate in red and sort of GOE or like 2.5, 2.1 billion year old sulfate in the yellow. The only thing I want you to pay attention to is the fact that you can see the yellow boxes below the red box. And a prediction of the the system I set up here is that because we need ozone photochemistry to drive these negative values in O2, this offset seems to conform to this prediction that in an atmosphere without O2, we should see sulfate taking in sort of crustal looking like values, triple oxygen. Whereas once we have oxygen in the atmosphere, we start to see O2 with this negative signature, which meets our prediction. Okay. Okay, now we're going to move to the mid-Proterozoic. Okay, at about 1.4 billion years ago. All right, so just to go back to this diagram here, now that we have our sulfate values from geologic record that it evaporates, now we're going to project to what the tropospheric value may have been. Okay, so we're going to do that with just an experimental calibration of looking at how much of the oxygens in sulfate originates in O2 versus water. Okay, so we take our data here, we're going to grow the y-axis because we're going to project down. So that bunches up our sulfate record. And then we project down to what our inferred O2 value should be. Okay, so that's what's represented in the green boxes here. All right, so now we have our O2 value. Now we want to actually think about what was the productivity contribution and what was the stratospheric contribution to this setting this O2 value. Okay, so we're going to the 1.4 billion year old Sibley group. This is in Ontario, just north of Lake Superior. And the reasons for going here were a fewfold. So one is there was a lot of drill core because this was an area that was highly prospective for uranium mining a long time ago. They found sulfates, so check, that's good. But the main reason is that this is interpreted as a terrestrial environment or an old lake. And why that's important is that it's generally thought, although this is a little bit controversial, that the biosphere at this time was largely in the marine environment. There wasn't forests, we know that, but it's thought that maybe the terrestrial environment had very limited biosphere at that time. What that means is that this might lead to much less organic carbon being delivered to terrestrial environments. We need organic carbon for things like microbial sulfate reduction. And so that might actually lead to better preservation of these triple oxygen isotope signatures in sulfate than in the marine environment. Because as sulfate turns to sulfite, we can get O exchange with water. If we prevent that reaction in a very organic matter limited environment, then perhaps we can preserve these atmospheric signals much better than a marine environment. And sure enough, this more recent work with Anna, we can see that if we look at these terrestrial samples at the top, which I'll talk more about in a little bit, and then we walk through things that are sort of considered restricted marine, so you can think of maybe ponds somewhere that get intermittently flooded, to semi-restricted settings where we have sort of a lagoonal setting or an evaporite basin that was seawater that was isolated from the ocean for a while, and then all the way up to hydrothermal systems, which are only sourcing their sulfate from seawater. We can see that we see this loss of triple oxygen isotope values as we move from this more terrestrial to marine setting. And all of these deposits sort of overlap in their age uncertainty of about 1 billion to about 1.3 billion years ago. All right. So to sort of back out what this O2 flux would be, we're going to constrain what atmospheric chemistry was. We're going to add in the impacts of isotopes. So we're going to need to know how these oxygen shuffle around when we change CO2, ozone concentrations, things like that. We're going to assume modern stratosphere tropospheric exchange. There's not really a proxy for this, but when people have looked at this parameter over very different atmospheric states, doesn't seem like it's nearly as big of an impact as changing O2 and CO2 concentrations. And we're going to vary the amount of O2 incorporated into sulfate over this experimentally determined range. Okay, so we're going to put that together in a simple model and then do a simple Monte Carlo approach to get an uncertainty on our ultimate estimates. Okay, most of these things have been calibrated in the lab, just looking at changing O2, CO2 concentrations in little reaction vessels and looking how isotopes shuffle around. But I just want to focus on O2 because that's definitely one of the most controversial inputs that we're putting in here. So what was the concentration of O2 over the Proterozoic? It is a very controversial topic. It certainly led to a lot of hurt feelings, friendships being broken apart, I think. But it really comes down to one of these really big fundamental questions. Like if we were to toss a rabbit back in time a billion years ago, like would it suffocate or not? And and it kind of comes to this question like, did animals have to wait for the environmental conditions to be perfect for them to evolve, or did the environmental conditions for animal life precede it by a long stretch at the time and sort of this evolutionary sluggishness? But we do have some intuition without fancy geochemistry. So we know that we're in an atmosphere that has some oxygen in the protozoa. So we can say that we're above Archean levels. Yeah, don't have time to talk about how we proxies for Archean O2. If we walk up a little bit from about 100,000th of the modern inventory to 1,000 of modern atmospheric O2 levels, this is where people have predicted that the atmosphere will be a bit unstable. And this is largely because we start to hook the greenhouse on methane. And so because under really low O2 levels, we limit how quickly we're going to oxidize methane in the atmosphere, meaning that it can become a much more important greenhouse gas contributor. And then it just takes a little puff of O2, we oxidize all that methane, and then maybe we tank the earth into snowball Earth, which is inconsistent with the sedimentary record at this time. Okay, so let's say that we're above 0.1% modern O2. This is also where folks like Jim Casting at Penn State have calculated that this is where we really get a resilient ozone layer as well, which we need for these triple oxygen isotope signals. So let's assume that we're above 0.1% O2. On the high O2 end, when we look through the Proterozoic record, we see this complicated picture of redox. So margins today that are fully oxygenated right down to basement. At this time, we see this complicated look of ferruginous, iron-rich, and acidic waters. And so we can say that, like, how much O2 do we need in the atmosphere for fully oxygenated water columns? And if we don't see that, we must be below that level. And folks like Don Campiel have put estimates out around 10%. There. And now in between this is sort of where we're stuck, which isn't super helpful because it's also within this range where animal life could exist. And so this is where a lot of controversy is. So people have thrown tons of geochemical proxies at this, and there isn't really any consensus to date. So we can separate this out in sort of the high camp and the low O2 camp. And look at what the consequences of these different views of the Proterozoic are on productivity with our triple oxygen isotope system. Okay, so in the high O2 world, say we go from 1 to 10% O2, it would predict that we basically have 41% of modern productivity, well, which is basically modern marine productivity. And in the low O2 case, we're about 6% of modern productivity. Okay, again, that's just saying that this O2 flux from the biosphere is about 6% of what it is today. Okay, now, we can't conclusively rule out either one of these scenarios, but the first case where we have a high O2 world in the Proterozoic essentially says we take the modern Earth, bulldoze off the terrestrial biosphere, we're back in the Proterozoic. The low O2 case says we have about 15% of modern marine productivity, and perhaps this is consistent with ideas of nitrogen limitation. If we had more metal depleted oceans, maybe synthesizing things like nitrogenase would be tough if we had like molybdenum and nickel limitation or vanadium limitation in the ocean. Maybe we have issues with P limitation because of less oxidative weathering, things like that. Or maybe it's just some things baked in with a less complex biosphere that makes it a bit less resilient. I don't know. But I think there's a lot more, a low O2, low productivity world, certainly more consistent with a lot of ideas about the Proterozoic, but to be continued. Okay, now let's look at the end of Earth's great oxidation event. Okay, so this is about 2.4 to 2 billion years ago. That's sort of how it's defined by folks like Dick Holland. But this is when Earth first got its oxygen, and it's inferred from a lot of different proxies that maybe we were in this transient period of very high O2 levels across this interval, which a lot of geochemical proxies point to. Okay, so these are results that are coming from the Belcher Islands of Hudson's Bay in Canada, and this is what they look like in space, which is really cool, I think. And so you have this folded terrain where you kind of weathered off the top, and it leaves this really beautiful imprint here. And so these are sort of a special set of rocks that are about 2 billion years ago. There's not really many well-preserved sedimentary environments of this age. So this was a nice place to work. Here's just a pretty picture. And then work by my colleague Malcolm Podgkis did a really good job of dating some of the underlying strata in the stratigraphy here. And so we were able to assign dates, some of these barytes that we found in stratigraphy, to about 2.018.5 billion years ago to 2015, which was great, which puts us right at the end of Earth's great oxidation event. And through some other geochemical gymnastics with barium isotopes and other things, we were able to put forward arguments that the sulfate is really coming from terrestrial environments and that the barium was coming from the marine environment. And so that was neat. Okay, but the main point is that we see this huge drop in the triple oxygen isotope values right at the end of the GeoE. So here we have all these evaporites that are and some caps that's preserving these negative values, but not super negative. And then we have this huge drop basically right at the end of Earth's great oxidation event. So what's going on there? So there's two interpretations of this. One is that for some reason, formations during the GeoE, we're just getting a smaller contribution of an atmospheric signal into sulfate. Okay, maybe that means that there was more microbial sulfate reduction in these environments where we're preserving these signals. I don't know. But I think this is a little bit unlikely because we're sourcing so many different formations over hundreds of millions of years in very different parts of the planet. So I think that that sort of general statement on a local level is a little bit tough to support. An alternative interpretation is that we're actually seeing real-time changes in major components of surface Earth. And so being agnostic of O2 concentrations at this time, if we just interpret these triple oxygen isotope results and think about productivity, we can say that even if we have no change in O2, so we ignore all of the redox proxy stuff and we just say, let's keep O2 constant at somewhere between 0.1 and 1%. Just based on the triple oxygen isotope values, it suggests that we need at least a 5-fold decrease in the overall productivity of the biosphere. If we had this major dip in O2 levels, which a lot of people have inferred, we could have as much of a dip in productivity of about 99%. So that's not saying that we're having extinction because we can't actually say that certain organisms went extinct. We don't have that ability with microbes this far back, but it seems like a lot less life. And based on our best geochron that we have available, it seems this shift happened in much less than about 40 million years. All right. Now let's move to the Phanerozoic, and this is some really cool work that Anna's been up to. And so when we look at the triple oxygen isotope values of evaporites, and these are marine evaporites, which is important because as we look through the sedimentary record, one of the most difficult things to control for is depositional environment and create like a consistent thread as we compare modern environments to sort of old ones to kind of old ones to really old ones. And marine evaporites are neat because we know that they must be forming in warm places. We know that they're probably forming in sort of near equatorial latitudes. And so it gives us some consistency in terms of what we're comparing through time. And what Anna found is that there's this shift that we see from about 480 to 432 million year old evaporites up to 320, 307 million year old evaporites. So what could ultimately cause this? Well, it could be a lot of things. It could be an increase in productivity, increase in PO2, a decrease in CO2, a decrease in the amount of O2 getting into sulfate. But the cool thing is that I think all of these are consistent with the rise of land plants. And so here, even though there's a lot of different proxies that deal with the O2 cycle, here we have one of the biggest changes that ever happened on the planet, which is the major colonization of the terrestrial realm by green things. And here we see an imprint of that in the atmosphere, which is really, and I encourage you to talk to Anna about this because there's a lot more interesting stuff here. So now it's going back and thinking about just some of the predictions we laid out at the beginning of the talk. Okay, so we shouldn't see an appreciable 17O signal in atmospheres with no oxygen. Okay, I think we did that. If O2, CO2 productivity have changed, then this should be reflected in 17 records of atmospheric O2. I think we see big changes. And if we know 2 out of 3, we should be able to solve for the third. And so I think that the record conforms to that, that we can make these predictions. Okay, so now let's take a step back to the overall record and see what happens when we actually run through these sorts of treatments through every sample. Okay, and we can produce a record of productivity through time like this on the upper axis. So this isn't just the triple oxygen values, this is actually pooling together every productivity estimate that's being put forward throughout Earth's past with different tools, in different models. Okay, and then this is just put on the backdrop of when we think different types of life were around. And so we see largely this monotonic increase in productivity through time, but there's a huge amount of uncertainty by orders of magnitude, which I think is really important to bring home, is that when we're looking at papers where people are making very, very concrete claims about how the carbon cycle was operating at a certain period of time, you know, and the best we can do over, let's say, the Proterozoic is a couple orders of magnitude variation, maybe over broad intervals. You know, that's a huge uncertainty that just needs to be accounted for. But even with this uncertainty, now let's put forward some fun numbers just to everyone can go home with. So here's some basic questions I began to talk with. How much carbon's ever been fixed? How many cells have ever existed? What's been the most abundant organism? Extinctions, what impact have they had? And then where's the majority of the uncertainty, just as sort of marker for where we should put a lot of effort in the coming years. And we can start to put answers to these questions. So how much carbon has ever been fixed by the biosphere? We can estimate that about 10 to the 11 to 10 to the 12 billion tons of carbon has ever been fixed. That's a big number that may not mean anything. But if we take, for example, that the Earth contains about 10 to the 9 billion tons of carbon in it, that means that the biosphere has cycled through this whole, every atom of carbon on the planet, something like 100 to 1,000 times. Now, of course, we don't have access to all of that because apparently people think a lot's in the core. But if we look at estimates from folks like John Hayes about how much carbon's actually reached the surface, suggests the biosphere cycled through this like 1,000 times. Also interesting to think about is that if oxygen photosynthesis never evolved, and when we look at just the productivity bumps that that metabolism gives us, as we move out into exoplanet land, and we think about what happens if we don't evolve this metabolism, at least on Earth, if this never happened, and we just take Archean productivity estimates and just take them up till today, then the biosphere probably would have only cycled through this carbon stock of 10 to the 9 billion tons of carbon once. So that's just sort of like an interesting way of, I think, framing how important oxygen photosynthesis is. How many cells have ever existed? We can estimate that more than 10 to the 39, but less than 10 to the 41 cells ever has. And when we project forward with where the planet's going, in terms of as the sun gets brighter, the Earth's going to get warmer, in about 1 billion to 2 billion years, the oceans will boil and everything's going to die. And if we project these productivity values forward and sort of put in some markers for when the terrestrial biosphere will likely die, and then eventually parts of the marine realm, we can estimate that in Earth's entire history, it's probably only ever going to have around 10 to the 41 cells. And that's sort of like the maximum capacity of our planet, which is an interesting benchmark, I think, when we're comparing Earth to other exoplanet bodies out there. What's been the most abundant type of organism? It's almost certainly cyanobacteria. Even though they're not today, they only take up about 10% of modern productivity. Things like SAR-11 are more abundant in the ocean. But as we move back in time and we move into regimes where cyanobacteria dominated global productivity, it actually, over time, their abundance actually outpaces anything else that could have existed. What impact have extinctions had? One could say basically nothing for this overall trajectory of life on the planet. And insofar as these like really high stress events, like smacking an asteroid into the planet, allow for new niches of life to take footholds that weren't previously possible, one could make an argument that maybe these death events actually led to an overall increase in life. And where's the majority of the uncertainty? It's definitely Earth's great oxidation event. So that's all I've got for today. So thank you all very much for your attention, and happy to take your questions.